Hi everyone. Today we're going to talk about missense mutations, one of the most common types of genetic variations identified in humans and patients. Over the past few decades, I've had the opportunity to work closely with biologists and clinicians, investigating these mutations in detail. A missense mutation occurs when a single nucleotide change in DNA leads to a codon that codes for a different amino acid. This might sound like a small change, but its impact can be profound. It can alter the protein structure, its function, or both. And of course, structure and function are deeply connected. When the structure of a protein changes, its function often changes too. So, one of the key questions we always ask is, does this amino acid substitution, the one identified experimentally through sequencing, actually affect the structure and/or function of the protein? And if so, does that help explain the patient's phenotype? To study further missense mutation or point mutation, one can use a variety of computational tools designed to predict the impact of amino acid substitutions. Among the well-known classical methods are tools like polyphen and SIFT. For example, SIFT somewhat evaluates how conserved an amino acid position is across evolution, the idea being that substitutions at highly conserved sites are more likely to disrupt protein function. Polyphen, on the other hand, combines sequence conservation with only some structural information to estimate whether a substitution is probably damaging to the protein. More recently, AI-based methods such as alpha missense have leveraged protein structure prediction to assess pathogenicity with greater accuracy, and models from the ESM family use protein language models to infer the effects of amino acid substitutions directly from sequence context without requiring explicit structural data. There are also meta-predictors that combine different approaches to improve reliability and overall prediction performance. Overall, there are well over 100 computational methods available for predicting the impact of point mutations on protein structure and function. You can explore many of these tools on my website, vls3d.com, in the Computer Tools section. Many are listed there, but obviously not all. When I work with my colleagues analyzing mutations found in patients visiting the hospital, we usually have only a small number of variants to assess. In these cases, I use the computational approaches discussed earlier, but I also perform detailed structural analyses with additional tools to better understand the potential impact of these point mutations. One recurring challenge, however, is how to report and visualize these results, since they come from multiple computational sources, and because a single mutation can affect many aspects of a protein. It might create a destabilizing cavity, interfere with the catalytic site, or modify a glycosylation site. To address this, it would be beneficial to apply a consistent set of methods and a unified style of data visualization regardless of the specific pathology. A few years ago, I developed a visualization concept for this purpose. This is to investigate one point mutation at a time, and I called this plot the flower donut traffic light plot. The diagram resembles a donut with petal-like segments forming a flower shape, and it uses a red, orange, and green color scheme, like a traffic light, to indicate the predicted impact of a mutation. Green for neutral, orange for moderate, and red for likely major effects. In this episode, I'll explain the idea behind the flower donut traffic light plot and how it could help standardize the way we represent variant effects. This is just a proposal, of course. Before we start, I want to emphasize that this video is educational. The computational analyses we discuss can help explore how mutations might affect proteins, but alone they are not diagnostic tools. Predicting the medical or clinical impact of a mutation requires expert investigations. There are many reviews in this field. I've written some myself, but I'd like to highlight 4 for reference. These papers discuss several key concepts regarding how point mutations can affect protein structure and function. One of them, from Carell's team, is particularly noteworthy. They coined the term biostructural pathology to describe this area of research. Although the term isn't widely used today, I still find it very insightful. Their examples involving the serpin family and the many diseases associated with mutations in these proteins are especially illustrative, not least because serpins are fascinating. They adopt a metastable native conformation, meaning their folded state is not the most thermodynamically stable one. This allows serpins to undergo a large conformational change upon protease binding and act as very efficient macromolecular inhibitors. The other reviews are equally important, offering a broad and insightful perspective on the field, with clear conceptual overviews. Now, one may wonder, what exactly are the questions here? There are already many computational tools to assess the impact of a point mutation, so why spend more time on this? Well, it's not that simple. With most prediction engines, you'll often end up with a conclusion such as this mutation may be damaging. But is that really enough for the patient and their family? Probably not. And if we want to understand what's happening at the molecular or even atomic level, especially if we start thinking about developing treatments, we definitely need to investigate much more deeply. A single amino acid substitution can affect many aspects of a protein's structure and function. It may alter stability, flexibility, and directly or indirectly intermolecular interactions all at once. So how do we represent or plot all that in a clear and meaningful way? This type of plot can't condense every piece of information, of course, but it could still aggregate many key results in a single visual summary. After experimenting with different visualization strategies, I eventually developed the flower donut traffic light plot. To build a flower donut traffic light plot, we first need to define the key category of impacts that a mutation can cause. I propose 4 main types. Then, for each category, we compute various parameters and, depending on the results, we decide if the mutation has a strong, moderate, or neutral effect on these specific parameters, which we then color in red, orange, and green, respectively. The first category relates to intermolecular interactions. A mutation might perturb interactions with another macromolecule, a membrane, or a small compound. A mutation could also alter a post-translational modification, for instance, allowing or preventing interaction with another protein or an antibody. All these can fit within the intermolecular interaction category. However, it's important to remember that these categories are not strictly separated. For example, if a mutation prevents glycosylation, this could also affect the stability of the protein. So, it's never a simple 0 or 1 situation. Several effects may overlap, and we must keep that in mind when annotating the other categories. Many different computational methods can be used at this stage to investigate or predict intermolecular interactions. The next category covers protein dynamic effects. Here, we want to understand whether the mutation changes local flexibility or affects global domain motions or allosteric transitions. Again, various computational tools can help us explore these aspects and can be done in 2D using only the amino acid sequence or using the 3D of the protein. Another category is structure and stability. These include many molecular mechanisms or parameters such as breaking a disulfide bond, creating an internal cavity, or computing delta-delta G to evaluate whether a mutation is stabilizing or destabilizing. Several computer-based approaches can be applied here as well. And finally, the last category, called here Other Key Molecular Determinants, is particularly critical. It includes information about the solvent exposure of the wild-type residue and its evolutionary conservation, 2 features that often provide strong clues about the impact of the mutation. Here again, many computer methods can be used to estimate these parameters or properties. Along with performing various computations, it's essential to use interactive graphical tools to verify algorithmic predictions. thus a human-in-the-loop approach to ensure critical evaluation rather than blind reliance on automated outputs. In fact, many tools are trained on databases that contain many annotation errors, so it is better to double-check. Here we can see the proposed 4 categories and the associated parameters that I use to make the plot. The same mutation can obviously impact different categories and parameters. For each category, many different computational approaches can be used. For example, delta G values can be computed in several ways, either in 3D, using a protein structure, or in 2D, relying only on the amino acid sequence as input. Large protein models can be used to explore stability, and saturation mutagenesis can be performed in silico by systematically replacing each wild-type residue with the other 19 amino acids. This type of analysis can also be done using 3D structural models. To study flexibility, one can use molecular dynamics, simulations, coarse-grained methods, or even AI-based methods that predict dynamic properties directly from sequence. It is wise to use several approaches and compare. Further, it is relatively easy to compute solvent accessibility using any modern 3D visualization program, but this property can also be estimated relatively well using deep learning-based predictors. Similarly, secondary structure elements can be analyzed from 3D structures with standard molecular modeling tools or predicted directly from sequence, usually with very good accuracy. Many programs can evaluate hydrogen bonds, cation-pi interactions, or salt bridges. However, it's important to pay attention to the distance thresholds used in these packages. If the protein structure has low resolution or comes from a model structure, these threshold values may need adjustment. In such cases, it's often advisable to soften the threshold values to avoid missing key interactions. Here is an example of a flower donut traffic light plot. In this example, 2 petals are highlighted in cyan. These correspond to 2 critical parameters: the solvent exposure of the wild-type residue and its evolutionary conservation. Both are essential to understand the potential impact of a mutation. Most disease-associated mutations tend to involve buried and highly conserved residues. This type of substitutions often destabilize the protein and can also alter its function. When problematic mutations are not buried, they often appear in binding sites where they can disrupt interactions with other proteins, DNA, or small molecules. And then, with this plot, we go step by step on all the other parameters discussed earlier, category by category. In this figure, the color of each petal reflects the impact of the computed parameter. Red indicates a strong effect, orange a moderate one, and green suggests a neutral or minor effect. In the example shown here, the residue is highly conserved, so the petal is red. It is partially buried, giving an orange color for solvent exposure. And because this mutation does not affect a disulfide bond, that petal remains green. Next, imagine you have 2 or 3 mutations to analyze. By simply looking at such plots, it becomes relatively easy to compare them visually. This is it for today. I hope the concept we discussed will be useful for your projects. If you have other types of plots or visualization ideas to suggest, feel free to leave a comment. Possibly radar plot, heat map matrix, stacked impact profile, or 3D radial bubble plot could do it as well. Something to explore. And until next time, take care.